Chapter 23 starts a new section of biochemistry, where we look back at the three major biomolecules, carbohydrates, lipids, and proteins, and try to understand how they're used for energy and synthesized in our bodies. Because the end parts of metabolism, the citric acid cycle and the electron transport chain, are generally shared when breaking down all three biomolecule types, we'll talk about these final and central parts of metabolism that begin with a molecule acetyl-CoA before we talk about how we get to acetyl-CoA from each type of biomolecule. In this chapter, we introduce the concept of metabolism, really just any of the reactions that our bodies do. Sometimes the reactions will be part of processes that take energy to make larger molecules that store energy. Can you think of an example of a large carbohydrate or lipid molecule that we might use to store energy? We call these processes that make larger molecules anabolic pathways. When you break down a large molecule to use the energy, this is called catabolism, and the sets of reactions that accomplish them, catabolic pathways. A pathway sometimes is linear, where we completely convert the starting material to a product, or sometimes they're cyclic, where the set of reactions give us back the original starting material, but convert a few atoms into energy or waste. Examples include the citric acid cycle and the urea cycle, or in plants, the Calvin cycle. Metabolism happens in several areas of the cells, and sometimes this leads to important features of metabolism. Most DNA metabolism occurs in the nucleus, as we saw in the last chapter. Most protein anabolism, remember this is building up a protein from the amino acids, occurs in the ribosomes, which are scattered throughout the cytoplasm of a cell. We also saw this in chapter 22. When we talked about proteolytic enzymes in chapter 21, we talked about how most protein degradation, which is catabolism, that occurs within a cell occurs in the lysozyme. Most of our central metabolism occurs in a specific organelle within the cell called the mitochondria. Mitochondria have a structure that is an outer membrane and a separate inner membrane. The inner membrane holds most of the enzymes responsible for the electron transport that will harvest energy to be used to make ATP, including ATP synthase. Between these membranes, there's an inner membrane space. The inner membrane has all sorts of folds called cristae. Why might the cristae be important to mitochondrial efficiency? The cristae increase the surface area of the inner membrane and allow for more enzymes in the same space. There are several molecules that we'll see turn up over and over and over again through the next 4 chapters. These molecules are some of the most important ways that energy is developed and used within a cell. Whenever we see these molecules, we want to pay particular attention because they help us to understand when energy is being used and when it's being produced, and how the pathways are connected. We'll talk about each group of molecules separately, then return to this summary slide. The first set of molecules are the ribonucleotide phosphates. Notice that these are simply ribonucleotides with different amounts of phosphate. The ribonucleotide portion gives an easily recognizable structure that an enzyme can bind onto. The phosphates are the major way that the energy is stored in our cells. Any ribonucleotide can store energy, but adenosine is by far the most common. Adenosine triphosphate, or ATP, is the major energy currency of cells. We're going to think about all of the energy that we talk about in ATP equivalents. Whenever ATP energy is spent, it loses a phosphate. It is most common to lose a single phosphate, which would give us adenosine diphosphate, or ADP, which we think of as the equivalent of spending 1 ATP. If we lose 2 phosphates, which is less common, we get adenosine monophosphate, AMP. We consider this as equivalent to spending 2 ATP. The next set of molecules are the flavins, flavin adenine dinucleotide, or FAD, and dihydroflavin adenine dinucleotide, FADH2. The general form only differs at the flavin, which you can see shown at the bottom, and the rest of the molecule is just to give an enzyme something to recognize. These molecules are related by redox. FAD is the oxidized form, and FADH2 is the reduced form, which means that these tend to be partners to other redox reactions on molecules. You can see the 2 forms of flavin again at the bottom. Because flavins are able to exchange 2 hydrogens at once, they are most likely cofactors for all reactions converting alkanes to alkenes, where FAD is used and FADH2 is produced, or alkenes to alkanes, in which case FADH2 is used to reduce the alkene to an alkane and FAD is produced. The nicotinamides are also redox cofactors, NAD+ being the oxidized form and NADH being the reduced form. Again, the only change in the structure occurs in the nicotinamide, with the rest of the molecule being a place for an enzyme to bind. Notice that for all three cofactors, the enzymes are recognizing an adenosine. When NAD+ is used, it catalyzes the oxidation of polar bonds, most often an alcohol or an aldehyde. NADH is produced in these reactions. Conversely, NADH catalyzes reductions of polar bonds and produces the more oxidized NAD+. Coenzyme A, often abbreviated COA-SH or COA-SH, is responsible for carrying acyl groups, like in fatty acids, and in this chapter specifically acetyl groups, a 2-carbon acyl group. Notice that again adenosine is part of the structure. Acetyl-CoA is the entry point into the central metabolism. Again, our important molecules that we want to keep track of are all forms of ATP, which are used for most energy functions in the cell, oxidized and reduced forms of both FAD and NAD+, and acetyl-CoA because it can be metabolized in the citric acid cycle and the electron transport chain and converted to energy. ATP is the major energy currency of the cell, but phosphates carry energy in many forms. The phosphates on the left are actually higher in energy than ATP, so can readily phosphorylate ADP and make ATP. The phosphates on the right include ATP and ADP in similar molecules, and lower energy phosphates that can be made from ATP. Our goal is often to make the phosphates higher in energy so we can collect its energy as ATP. We'll see this in the citric acid cycle, but even more in glycolysis next chapter.